get started. Good morning, good afternoon. Welcome to Masterclass Live for metabolite identification with mass spectrometry. My name is 𝗏𝗏𝗏𝗏𝗏𝗏𝗏 and I'm the Market Development Manager for Pharma at SCIEX. I'm very excited to be here with all of you today. This masterclass is a great opportunity for all your curious minds out there who want to learn more about MS-based solutions to identify and characterize metabolites throughout the drug discovery and development process. Rahul Baghla is a research scientist with 12 plus years of experience in various applications of mass spectrometry, delivering in pharma, biopharma, and CRO industries. So you guys are in great hands. He specializes in implementing new mass spectrometry technologies in bioanalytical and analytical method development, validation, drug discovery, ADME assays, and clinical studies. He has supported various functions within pharma industry, including nutrition, wellness, and oral care-based clinical trial studies over the years. Rahul has a master's degree in pharmaceutical analysis, master's in business administration, and a professional diploma in clinical research. He is responsible for continuous engagement with pharmaceutical community to identify major analytical challenges and provide solutions and find significant analytical opportunities for SCIEX. So my friends, get ready for an adventure that will challenge your minds, expand your horizons, and turn you into true masters for metabolite identification with mass spectrometry. Rahul, the floor is yours.
Thank you, Bahar. Hi, everyone. Welcome to the masterclass today on metabolite identification from beginning to the end. So in today's masterclass, we will split it into two parts. We'll have a basic presentation that will cover around the basics of the instrument and the basics of the software. And then we'll go live on the instrument. And then we're going to tell you how to set up the instruments and how to set up the mass spec methods and how to set up the software for processing data. So the instrument we are going to use today for our masterclass is the SCIEX XenoTOF 7600 system. It's a benchtop QTOF with the new alternate fragmentation technique, which is called electron-activated dissociation, which is actually really important when you are dealing with all your metabolite identification or you're trying to identify the site of biotransformations that we're going to discuss in the upcoming 60 minutes probably. So yeah, before again, thank you very much for joining. And meanwhile, I will go back and set up and bring up my presentation maybe in a minute. And all right, I'm all set. All right, guys, thank you everyone. So the agenda today that we're gonna cover is for this workflow is we will start with the introduction to MetID workflow and the importance. And then we'll have a brief session on introduction to SCIEX solution for MetID, which will be introduction to XenoTOF 7600 system, and along with the introduction to the molecule profiler software. And then we're going to talk about the experimental conditions, pretty generic conditions that I use for MetID experiments with overview of acquisition method and the data processing with molecule profiler. Both will be live on the instrument PC. And then we'll have a data review with the results from the molecule profiler. And we'll close the class today with the conclusion and some Q&A at the end. So talking about MetID, right? So MetID and importance. So we know that drug metabolism characterization is essential for optimizing pharmacokinetics and pharmacodynamics, and also the safety profiles of drug candidates in the drug discovery and development process. So as we know, the drugs must reach the site of action to elicit their pharmacological effect after the administration. And it's very important to have a stable drug, right? If they show like an inferior PK properties, such as high clearance, short half-life, or low bioavailability. So the PD effects are likely to be suboptimal for these drugs. So it is very important to identify the site of biotransformation or where the metabolism is happening. So in vitro metabolism studies in human and animal tissue preparations are one of the very valuable tools for identifying major metabolism pathways. Moving to the next one on the current challenges for MetID, we know that MetID is being done for years, and it's very important phase as we discussed in the previous slide when we talk about the drug discovery process. So one of the major challenges that we see with the MetID experiments is the low abundance of the relevant circulating metabolites, right? So when the drugs are exposed to the media or to the body, we know that there are multiple metabolites are formed, and the concentrations of those metabolites are varies, and it is very hard to generate a good MSMS spectra for low abundant metabolites. And second is the ionization efficiency in the complex matrices. This is usually taken care by LC separations, but that leads to really longer run times. But now with the UPLC, since we can have a shorter run times and the sharper peaks, and then separate those interferences so that we don't see matrix separations as possible. But at the same time, we need the mass specs that have a high speed, high acquisition speed, so that you get like a low cycle times and still get enough data points across the peak if you have to use that data for quantitation purposes. The third is on the qualitative information. We know the CID, which basically sometimes it lacks the qualitative information or doesn't have enough information, especially when we are talking about phase 2 metabolites. So that is being overcome by EAD with the XenoTOF 76 system. And then also in terms of if there are hydroxy metabolism, right? So it's very important to have like an isomeric conformation where actually the hydroxylation is happening. So for that, you need a richer MSMS spectra, which is not possible with CID sometimes. So we have additional complementary technique that you use, EAD, that you use for generating much richer MSMS spectra. Especially the case, for example, another case for the phase 2 conjugates, where the CID fragments basically doesn't help much to localize your site of biotransformation. For these type of conjugations, they are basically relies on accurate mass and observe neutral losses. For example, for glucuronides, you only see the neutral loss of 176, and your MSMS spectra usually comes from the drug. So it is very important to generate much richer MSMS spectra where you contain the information from your conjugates so that you can identify the site of biotransformation. So coming to the SCIEX solution for MetID workflows, in the next few slides, we're going to talk about the XenoTOF 7600 system and the molecule profiler software. Coming to the XenoTOF 7600 system, as I showed you in the brief video in the beginning, so that's the benchtop QTOF system that talking about like the system advancements compared to our previous generation QTOFs. It comes with a XenoTrap. XenoTrap is basically is a small trap that we have introduced to improve your cycle time. So, and what is cycle time? We're going to talk about in the next slides and why it is important. But what it does is basically it improves your MSMS sensitivity 5 to 20x compared to the conventional TOF system. So it can be coupled with EAD and CID both. So it is the first point that we discussed in the challenges, right? The MSMS spectra for low abundant metabolites. This helps a lot there. We have a new Q0 design, which basically improves your ion transmission compared to the previous generation. And then we have electron activated dissociation. This is EAD that we have been talking about, right? So this is a different, what you call it, a fragmentation or the complementary fragmentation technique that we generate fragments with electrons. And that can help you to localize your biotransformation site because the MSMS spectra from EAD are more information rich. And the wide dynamic range detector. So the new detector, 5 GHz 10-bit ADC detector that basically gives you 5 orders of linear dynamic range in case you want to use accurate mass for your quantitation workflows. So what is duty cycle, right? So duty cycle is basically the percentage of ions injected into the TOF system. So in the regular conventional TOF systems, it is 5 to 25%. So basically you are with the conventional TOF, you lose 75 to 95% of the ions. So you have only basically 5 to 25% of the ions going to the detector. So why duty? The next thing is like why duty cycle is not 100%, right? So the you know that the quadrupoles are basically in the QTOF system. The quadrupoles are basically the continuous beam uh, system, and the TOF is basically a pulse measurement technique. So when your ions are traveling through the quadrupole as a continuous beam, when they reach the TOF accelerator, we do lose a lot of ions, and only few ions go up to the detector, and this process keeps on going. So what we did with XenoTRAP is we introduced like ion gates. So what happens is all the ions in the quadrupole are basically trapped and then they are released to reach to the TOF accelerator. So here what happens, like all the ions, they reach the TOF accelerator at the same time, and then they hit the detector. So we are actually, what we are doing is we are trying to control the ions here, and then we release them all together as a packet so that more ions reach through the detector and you get higher duty cycles. So by doing this, we actually were able to achieve the duty cycle of more than 90%. So the next is another important part. This is the second feature about the XenoTOF 76 system, the EAD, right? We discussed about this complementary fragmentation technique. So how it works is the free electrons in the EAD cell are captured by ions, and then they form high intermediate radical state, which further fragments. So the idea behind EAD is, the good thing about EAD is what we can do is we can control the electron kinetic energy. So going in from the electron kinetic energy from 0 to 25, you can see on this scale. With the 0, the EAD cell actually works as an electron capture dissociation, where you have this works more for your multiply charged peptides, or it generates better suits for the fragmentation of multiply charged peptides and proteins. And at the kinetic energy of 5, this EAD cell functions as a hot ECD, so hot electron capture dissociation, which is more suitable for generating your fragments from glycopeptides, disulfide bonded peptides. And then at the kinetic energy of 10, the EAD cell features that EIO, which is electron impact excitation of ions from organics. This is for singly charged molecules. So all our meta-ID experiments that we do for the singly charged small molecules, they optimize between 10 to 15 of kinetic energy. So the electrons, as I said, the electrons introduced with the different energies, they basically induce the fragmentation in different type of molecules. But with EAD cell, it gives you the flexibility to adjust your kinetic energy based on the molecule that you're working with. So that was on the 76. Moving to the software part, the data processing part, right? So we have a molecule profiler software that helps you to identify and characterize your drug metabolites. It is an integrated SCIEX software, can be activated and process data with the molecule profiler license. So the SCIEX software is used for acquisition and data processing for your XenoTOF 7600 system or any QTOF system from SCIEX. And then the molecule profiler is an inbuilt software, but you just need a license to get it activated, and then you see this tile on the software. And also it comes with the Explorer, where you can actually manually process or review your data. So it's a combination of two very powerful softwares within SCIEX to address your metabolite identification needs. Talking about the highlights of the molecule profiler software, is integrated into SCIEX as we discussed. It supports multiple applications, not only small molecules. You can also process your peptide oligos or ADC data, suitable for metabolites and derivative identification and characterization. You can also look for the possible cleavages. You can build your compound library, and then it has a comprehensive biotransformation database. We're going to talk about that in the live software session soon. It does based on the MS1 peak identification, and MS2, basically the MS-MS fragmentation annotation is used for structure elucidation. And it also can help you to do sample comparison when we call it like a correlation. So for this data set that we're going to discuss today, these are the very basic reverse phase conditions that we use. We do, darunavir was the drug that we studied or we're going to discuss in this class today. We incubated at 37 degrees Celsius in rat hepatocytes at the starting concentration of 1 micromolar. We use XenoLC80 system with Phenomenex Luna Omega Polar C18 column, 2.1 into 100 mm with a very basic reverse phase conditions with 0.1% formic acid and water and estronitrile. We operated a system in data-dependent acquisition. You might see it as an IDA in the methods in SCIEX. We did run it with the CID and EAD both. The CID conditions were with the collision energy 35 and C spread of 15 volt, very basic conditions to generate a good MS-MS coverage. And for EAD conditions, we had electron kinetic energy of 11 eV and electron beam current of 8,000 nanoampere in this case. The important part I want to discuss here, the chromatographic gradients. Usually, all your meta-ID experiments goes pretty longer gradients, but in this case, we did all this study within 6.5 minutes, so maintaining your good throughput. So this was, since it was a UPLC, so we were able to separate all the interferences and get sharper peaks within 6.5 minutes, and also the faster acquisition speed of XenoTOF 7600 system gave us enough data points across the peak for every assay so that we don't lose anything while doing this analysis. So that's basically the presentation part for today, quick. And then we're gonna move to live on the instrument PC when we're gonna discuss about the SCIEX OS, and I will show you how to set up the method, and probably we'll have a few minutes on the molecule profiler software as well. So let's move to the fun part, right? So that's SCIEX OS software. You can see here, this is a new software we have transitioned from Analyst, if you guys know SCIEX. So this has it's a very user-friendly, very intuitive software. You can see in the first, this is the first how the software opens up, right? The first interface that you see. So you have batch information on the front, then you have a queue, MS method, LC method. It's all like kind of tiled in terms of as a like setup. It's set up on the software as a tiles, and when you click on that, it's going to take you to that particular window. And then for the processing, we have Explorer, Analytics, and Molecule Profiler here. And in terms of management, your configuration includes your hardware profiles. And then library is in case you want to build library for your assays. And then event log is basically what's happening with your instrument. You record everything here. It's very intuitive, very easy to use. You can equilibrate your mass spec and LC right here. You can select the methods and equilibrate. You have a quick calibration checks right here, and then your device information always shows up here. When you click, it will exactly show you the condition of your mass spec or the LC condition that you're running in. All right, so I will start with the method. So I click on MS method, and then I click on the new, since I told you that we have used DDA, so that will be your IDA experiment here. So if I click on IDA, you can see it pops up a default inbuilt method. So we have different workflows assigned in each method. So it's a small molecule, peptides, intact protein, and glycopeptides. So in this case, since we're working with small molecule, if you select small molecule, it will give you all the default settings that we have embedded into the methods to make life easy for everyone. So source and gas parameters, after you set up your method duration, source and gas parameters are usually very basic based on your flow rate. So if you go to the SCIEX website, in the resources, you can find all the gas settings. I usually run my assays at around 0.5 ml a minute. So for that, I use very basic gas conditions with the GS1 and GS2 of 50, 60, current gas 40, and the temperature around 500 degrees Celsius for ESI. So GS1, just to let you know, the GS1 is a nebulizing gas and the GS2 is a drying gas. Then the curtain gas I prefer a little bit higher because the MET-ID samples are slightly complex matrices. So it's always good to have higher curtain gas to avoid any cross contaminations. So in terms of experiment part, you can see this is basically the IDA experiment with the positive polarity with a spray voltage of 5,500. The TOF MS start mass and stop mass are based on your molecular weight of your compound and what metabolites you're targeting. If you're conjugated metabolites, I would preferably ask you to keep like a little bit higher, more than 200 above your mass of interest or your parent molecule mass. And then we have reclustering potential. If you're working with the completely new molecules, I would prefer to keep it a little bit lower, around 40, 50 range, so that you don't have in-source fragmentation. Collision energy is 10 is fine because we are not fragmenting anything for your TOF MS or MS1. And the accumulation time is it depends. It basically identifies like how much is your peak weight, how much cycle time, or the total scan time you want. So in this case, for example, maybe we can switch it to 50 milliseconds here. And the second part is the IDA criteria, right? So here also we have a different inbuilt settings, small molecule, peptide, and metabolites. So in this case, I'm going to pick up the small molecule. Metabolites are basically for metabolomics application. So what it does, it changes your maximum candidate ions and intensity threshold default values. The maximum candidate ions is the probability of how many metabolites you are expecting to co-elute, right? So if you're using like a really short run times, there's a higher probability the more metabolites eluting at the same time. If you have a longer run times, then the probability is less. So I usually do is like maximum 6 candidate ions. So what it's going to do is it's going to trigger the MS for in case there are any coelutions for the 6 most intense MS1s that he's going to identify. And then intensity threshold is basically at what threshold you want to trigger your MS. That depends on the starting concentration or the how your molecule ionizes. But the beginning, you can probably start with 500 or 1,000 to focus on your low abundant metabolites. Dynamic background subtraction, we always keep it checked so that we don't see any background ions coming over and over. And then there's another option, exclude former target ions. So what it does is basically it helps you to set up that when you want to trigger MS. So, and then you don't want, and then you can decide that I don't want to trigger MS for one specific molecule or one specific mass for the next 6 seconds. So it also depends on how much is your peak width. So in most of the cases, since I don't know the chromatography, I don't know what will be my peak widths. So what I do is I just uncheck it and then just use a dynamic background subtraction. So for the MS, again, the TOF start mass and stop mass are based on what fragments you're expecting. I usually start with 50. And then declustering potential stays same like your previous experiment, and the collision energy and collision energy spread are basically to collect your good richer MS spectra. For example, in this case, in most of the small molecules, 90% of the small molecules, the collision energy of 15 and the spread of 15 give you good MS coverage. But you can change these based on your parent molecule. And then I'm going to show you, this is about the CID. And now if you want to go to the EAD, you have to show the EAD, click on the show EAD parameters. So that gives you an option to select your fragmentation mode. So you can select CID or either EAD. So once you select EAD, you will see the reaction time, pops up, and also the ETC is not option shows up here, the electron beam current and the kinetic energy. So kinetic energy is the part that we discussed in the presentation earlier. So this is the one that we have to use like between 10 to 15 for small molecule, and the electron beam current usually stays around 6,000 and 7,000. So my starting point is always like 12 and 6,000, and then you can play around a little bit if you want to see like richer MSMS spectra, but it varies from molecule to molecule. Other thing you can do is you can run in one MR-MHR experiment with the different kinetic energies. So in one injection, you collect all the data, and then you can pick up the best one for your ID experiment. For the reaction time, it depends how much you can use a minimum reaction time. I would say 5 milliseconds or 10 milliseconds. But the higher is a reaction time, is basically you're fragmenting more, right? And at the same time, my sweet spot usually stays between 20 to 30. So talking about accumulation time, usually 5 milliseconds are enough to clear off your EAD cell. So what you can do is whatever your reaction time is, you can add maybe another 10 milliseconds to the accumulation time, so that when the next ions gets into the EAD cell, make sure that it's cleared, and you have you get a clear MSMS spectra for your next ions too. So that's all on the MS part, how to set up your methods with EAD and CID with the IDA workflows. And then moving to the LC, I have Xeon LC here, where the interface looks like this. So you can define your flow rates and starting conditions with the gradient. Auto sampler, usually based on how stable your molecule is. I probably prefer 5 degrees Celsius here. And then also the column temperature is based on the chromatography. So this is just like a quick overview on LC and MS method. So then you can go to the batch. It's very easy interface on SCIEX OS. You type in your sample name, select your MS method, LC method, and then this rack type positions. These all comes from the LC that you're using, your injection volume, and the data file. Once you have everything, just hit on submit, and it's going to start acquiring your data. So once you have data, and then the second part comes is the molecule profiler software. So let me give you a quick overview of the molecule profiler software here. Since we discussed about the compound library, so you can see if you click on the compound library, it shows up the window that what you want to do. And then if you're setting up, you can look into the compounds. We have some inbuilt libraries as well, but if you are looking for a new compound, then you can just click on the new. If you're working on small molecule, you can select structure. If you're working on the peptide, you can select sequence. So in this case, it is structure. I'm gonna name it, for example, in this case, I have darunavir drug. I had so you put the compound name, open your structure here. You need to have a mole file to load it into the molecule profiler software. And when you click on the mole file, you can see it will draw a theoretical isotopic pattern. And it also gives you the chemical formula. And then with the experimental data, you can select your WIF file or the data file from the data that you have acquired, and it's gonna pick up the MSMS spectra for your molecule. So you can set it as a reference, and then when you click OK, it's gonna save your compound library. So once your library is set up, and the next is your processing parameters. So you click on processing parameters. This is how this window shows up. It shows already the method type is small molecule, but if you want to change, you can change right here. Small molecules, ADC, peptides, or oligonucleotides. So in this case, it's small molecule. I'm going to pick up the compound information from the library. We picked up darunavir. And so it will pop up the structure, will pop up the chemical formula, charge state, adduct, the M by Z. And then we have a peak finding strategies on the left. So if you're targeting the predicted metabolites or you're looking for all generic peaks that above a specific threshold compared to your control sample. You can also filter using mass effect, isotopic pattern, and TOF MSMS. And based on the MSMS, you can define like how many characteristic product ions you want to see. So as you keep on checking these boxes, your number of metabolite list will be keep on like going smaller. You will get much like true hits, but it depends what kind of workflow you're doing. So in the initial, for example, if I'm just looking for phase 1 or phase 2 metabolites, I just click on predicted. And instead of using any of the filters, my first attempt is to just process a very generic method, and then I can delete or I can discard the metabolites that I feel which doesn't make any sense. And on the right side, we have a biotransformations. You can select the sets, right? The phase 1, phase 2, phase 1, and it has like multiple options like GSH, biologics if you're working with the peptides. The good thing about this biotransformations is they are editable. You can add new biotransformations and you can remove some of them if you're not expecting them in your sample. So for that, you have to go to the biotransformations and you can make those changes. And then we have a chromatographic data where you have all the information about your LC. You can define your retention time windows, minimum peak width, minimum peak intensity. If you want to smooth the data, you can check this. And the good thing about this software is it's very intuitive. You don't have to go to the help section again and again. You just bring your cursor on the parameter and it will give you a small description that what it is for, right? For example, if I go to the MS parameter, if I click on M by Z tolerance, so it says type of minimum tolerance to find potential metabolites. So that means it's on the MS1 level to identify your metabolites. Same is the case with all other parameters too. And then you can also select your available adducts of the predicted metabolites, whether you're looking for ammonia adducts, sodium adducts, or just M plus H. You can pick here. You can perform background subtraction. If you're doing already in your MS method, you can skip here. Same as with the MSMS. You have MSMS tolerances, minimum peak intensity. For me, I preferably keep it around 100, and for minimum MS peak intensity should be around 500 or 1000, but it changes based on the data you acquire. And the source of reference MSMS spectrum, it will be you can select control or sample or the reference spectrum that you already have in the library. Same is for MSMS M by Z tolerance to compare the different potential metabolites, and also the fragment interpretation options, minimum signal to noise ratio, number of fragment peaks selected for assignments. So for example, in CID, we have 30 here, EAD, we have 60 default, because with EAD, you generate way more fragments compared to CID. And with EAD, you can break the rings. So you can check this box, break aromatic rings, and then the maximum number of bonds you want to break, you can select here. And you can do this after your data is processed as well. So it gives you enough flexibility. It's not like you set up something here and then you have to keep on reprocessing the data if you want to change this. So you can change these parameters in the final result table too. And then we have a formula prediction where you can put your elemental composition and also the tolerances for mass error. The confirmation scoring uses 4 different properties. You can define all these 4 properties, how much score you want to give, and then it's going to do the predicted metabolism. It's going to predict the metabolites based on this score from the different data files. And then in the compound specific parameters, you can decide in case you want looking for any cleavages. If you're not interested in cleavages, only metabolites, then you can select 0. But again, this is based on your workflow, what kind of experiment you're doing and what you're expecting. Mass defect numbers picks up automatically here when you upload your structure, isotopic pattern, and then the product ion and the neutral losses. So this is basically the spectra that we had in the library. But if you want to define, like upload a new file, you can do here as well. So you can upload your CID file and EAD file. And also based on what MSMS, looking at the quality of your MSMS spectra, you can decide the threshold and mass accuracy. So in this case, we have 5 millidaltons, and with the product ions, we are looking at about 5% of your highest intensity in this case. So this is a quick introduction to the processing parameters on Molecule Profiler software. And then the next is basically to process the data, right? So you click on the batch, and then you can select your data files here. So as I said, the Molecule Profiler software can process your CID and EAD data together, and it gives you like a quick comparison chart as well. So in this case, I've selected CID here, and then you can select the EAD data file in the additional MSMS data. If you have some controls like your blank samples, or you can select your blank samples here as well. And then pick up the folders where you want to save the result table, your processing parameters, and then apply options is basically if you want to assign structures to your metabolites, you check this box, and then type the result table file. For example, in this case, I have a T30 EAD versus CID. And then you just hit on the find metabolite. So this is going to start looking for the metabolites. It takes some time based on the filters in your processing method and also at your speed of your PC. So usually based on like my laptop configuration is like a SSD with 32 GB RAM, which is required to run SCIEX OS, or the SCIEX OS processing PC, I would say, or SCIEX OS acquisition PC. I have a same configuration on my laptop, and it usually takes maybe a minute or a minute and a half to process one sample set. But the good thing is you can queue up everything. You can import this as an Excel. You can import all the batch from your Excel sheets. And then, by the end of the day, you can just click on the processing and all samples will be done by the time you're back after a break or maybe in the morning. So it's very easy and medium throughput kind of workflow to address your drug metabolism needs. So since it's processing, I have some results in the presentation from the Molecule Profiler software. I can show you how it looks. So you can see here, this is the results pane from the Molecule Profiler software, and it gives you like the information on the potential metabolites, list of potential metabolites in the top, and then you have a EAD and CID MSMS spectra. So since you process both files together, you have basically both like as an inverted overlay. And those red labels are basically all unique fragments. So in this case, you can see with EAD, we see a lot of unique fragments compared to the CID. And especially they are important because they are maintaining or they are keeping the glucuronide structure, which is not possible with CID. So with CID, you see just a neutral loss of 176, and your MSMS spectra looks pretty similar to your drug molecule, whereas with EAD, you see a lot of unique fragments. It gives you the proposed formula structure information for all the match fragments, neutral loss information, and then predicted structure rankings with EAD and CID spectra weighting. So you can put the weighting here. So in this case, if you have 100% EAD, you can see it ranks the structure based on the overall score or the proposed assignments. So 1, 2, and 3. When you select 100% CID is going to use the MS spectra from CID, and it's going to reassign the ranks for your structure. And since you can see, you have a predicted metabolite structure here as well. This is a O-glucuronide, and this is basically different for each assignment, but this software also gives you the flexibility to edit the structure and assign it to MS if you don't like any specific structure. This is another example for the oxidation metabolite from darunavir. You can see the same interpretation pane. On the top, we have EAD, and the bottom, we have a CID spectra. You can see a lot of unique fragments with EAD, whereas they were missing in CID. So what it happens, this is like a completely easy software approach, so that you check the scores, like how much score you are getting based on the EAD and how much score you're getting based on the CID. So in this case, you can see the EAD score was much higher for this predicted biotransformation or cytobiotransformation, whereas CID was much lower. So higher score with the software is basically the higher is the probability of having that particular biotransformation site or the modification on the structure. So not only MedID, like you can use this software for your other workflows as well, like impurity profiling and the drugs. Coming to the next slide on the correlation analysis. So this is another important feature to do the relative quant. Molecule Profiler does it for you. So for example, in this case, the study of drug metabolism over time, we have time on the x-axis and the peak areas on the y-axis. So you can see in this specific, what we do is we process the all result tables that we generate with Molecule Profiler software to draw a correlation analysis. So in this case, you can see the darunavir parent, the concentration or the peak area was going down with time at the 120 minute compared to the 0 minute. Whereas the metabolite formation shows that, oh, definitely the drug is, the metabolites are forming in higher concentration over time. So we see higher peak areas with increase in the time. So this is really important if you want to have like a quick overview of the clearance profiles, right? And how the clearance is looking and how the metabolism is going. Quick overview of your a parent drug and as well as your major targeted metabolites. In this case, we have 2 glucuronide metabolites and 2 oxidation metabolites. And also we had time here. You can also have a different species if you're doing it in rat, human, or any other species. You can have a different species on the y-axis, and then you can have look across various species how your metabolite formation is happening. So the relative quantitation is basically, I would say, of all the drugs and the metabolites based on your MS1 peak or the TOF MS experiment. Yeah, coming to the last slide on the conclusion. Yeah, so we what we learned in the whole last 15 minutes, the data acquisition to analysis workflow was demonstrated using SCIEX OS integrated molecule profiler software with the XenoTOF 76 system. Electron activated dissociation, as we discussed, combined with the XenoTrap, definitely shows a great promise as a new fragmentation technique for delivering information that is definitely complementary to CID. A quick analytical methodology with just 6.5 minutes with a software-aided approach for metabolite identification was demonstrated to support high throughput workflows for the drug discovery process. I'm pretty sure this has been like pretty helpful for you, but still, if you need more information, you can scan this QR code and then it will take you to our website with more metabolite identification resources, or you can find some technical notes there. And then definitely on the acknowledgement, I would like to thank the SCIEX team, Jason, Kausan, Ishani, Nandita, Disha Thakkar, and Eva. To basically help me to set up all these experiments and helping me with the data, and also the Merck team, 𝗟𝗏𝗡𝗀𝗀, they provided us these samples for the sample analysis. So yeah, that's all from my side. We will switch it back to Bahar for any questions. Thank
All
you,
right.
guys.
Thank you. Thank you so much, Rahul, for the great presentation. Fantastic. Thank you again, Rahul, for a great session and for walking us through the details of MedID with 76. For those of you who attended, thank you for your participation. And if you have any questions, feel free to reach out to me or Rahul after the session. With that, I would like to conclude the meeting. Thank you.
All right. Thank you, everyone.